AS Level Biology 9700, Paper 1, Variant 3, October November 2025. And in this video, we will discuss from question number 14 to question number 26. So this is the second video on this paper. So we will start from question number 14. So the question number 14 is there. Which statement about phospholipids is the bilayer of a cell surface membrane is correct? The first option is the non-polar phosphate heads face outward and the polar fatty acid tails face inward. This is wrong because you know that the phosphate heads are hydrophilic in nature. They are water loving because they are polar, and the environment inside and outside the cell are aqueous. So these polar heads must face outward towards the water. And secondly, the fatty acid tails. So these are always hydrophobic, water hating. So because they are non-polar to avoid water, so they face inward, pointing towards each other in the center of the membrane. So according to this statement is not correct. So the polar phosphate head face in outwards and the non-polar fatty acid face inwards. So this is the correct option. Third option, which is C, again, it mentions that fatty acid tails face outward and the polar phosphate heads face inwards. Again, this is wrong. So we have to now the option D, the polar fatty acids tails face outwards and the non-polar phosphate face inverse. Again, they are showing that the fatty acids are polar, they are not polar. So B is the correct option of the question number 14. Now, question number 15 is there. And in question number 15, you can see they have given us the three statements and we have to choose the one correct option. Which descriptions of enzymes that choose the lock and key hypothesis are correct? The active site is complementary to the substrate, the enzyme act. Yes, this is correct. That is the active sites are always complementary to the substrate which is attached on it. Then bonds that form in the enzyme substrate complex change the shape of the active site. It cannot happen. This is very incorrect statement. It cannot happen. It is, and it change in the shape of the active site upon substrate binding describes the induced fit hypothesis, not the rigid lock and key model. So it does not happen in lock and key hypothesis. So most of the amino acids in an enzyme help in to maintain the specific shape of the enzyme. So this is also the correct statement because only a few amino acids make up the active site. So the vast majority of the remaining amino acids in the globular proteins are essential for folding, forming bonds, and maintaining the overall 3D tertiary structure. So that keeps that active site intact. So according to this, statement number 1 and 3 are correct, and this option goes to C, which is the correct option instead of all other options which are incorrect. Now, question number 16 is there, and question number 16 is the graph shows the result of an investigation into the effect of emalase on the starch at 3 different temperatures. So they have given us 3 statements. Which statements are correct conclusions using these results? So here you can see the optimum temperature is 50 degrees centigrade. At 50 degrees centigrade, you can see the optimum temperature is not 50 degrees centigrade. So it is less than 50 degrees centigrade. And the highest total concentration of reducing sugar in this specific, we cannot definite conclude it is precise optimum because we have been shown data for temperature between 30 and 60. So we cannot go with it. So this statement is not correct. The initial rate of reaction is highest at 70 degrees centigrade. Yes, you can see the curve at 70 degrees C. This it shows it is correct. The higher the temperature, the more quickly the enzyme denature. This is 101% correct. That is, when there will be high temperature, the optimum temperature will reach soon. So the enzymes will become denatured after getting their optimum temperature. So in this way, I would like to say the statement 2 and 3 are correct, and according to this, the D is the correct option. Now moving towards the question number 17, and question number 17 is there. The rate of enzyme catalyzed reactions in human cells is regarded. We have to be involved in this regulation. Again, three statements they have given us, and according to this, a change in enzyme concentration. Yes, this statement is correct. Enzyme concentration, the change occurs. So change in substrate concentration again, as the changing the substrate concentration indirectly affects the rate of reaction until the enzymes become saturated or the maximum is attained. So statement 3, inhibition by the final product of the reaction. So end product inhibition is a classic mechanism where the final product inhibits an enzyme earlier in the pathway to prevent overproduction. So according to this, three statements are correct, and the answer will be the A, option A, which shows these three statements are correct. So A is the correct option. Now question number 18, proteins in the cell surface membrane of human cells and mouse cells were labeled with fluorescent dyes. The human cells were labeled with the red dye, and the mouse cells were labeled with a green dye. Proteins in the cell surface membranes of human cells and mouse cells were labeled with fluorescent. So a human cell and a mouse cell were then fused to form a hybrid cell. They were fused. At first, the different dyes remained separate. After 40 minutes, the two dyes were evenly distributed in the hybrid cell surface membrane. What explains this observation? So again, they have given us four. All protein molecules in the cell surface membrane are fixed to cell structures within the cell, but phospholipid molecules move freely between them. This option is incorrect because it claims proteins are fixed. If they are fixed, the dyes would never mix. So this statement is not correct. And option B is incorrect because the proteins and phospholipids do not mix together in rigid attached groups like this. So again, this is not correct. Only protein molecules in the outer layer of the cell surface membrane can move freely between phospholipid molecule. It limits movement unnecessarily to just the outer layer. So again, this is not correct. Protein molecules in the outer layer of the cell surface membrane and those which span the bilayer can move freely between phospholipid molecules. So this statement is correct. Mixing occurs because membrane proteins are free to move laterally through the fluid phospholipid molecules. So this statement is correct. According to this, D is the correct option. Then move towards the question number 19, and question number 19 is, which statement about the cell surface membrane are correct? Again, they have given us four statements. So we will go with the first statement. Which channel proteins allow water soluble ions and molecules across the membrane? So this is correct because the hydrophilic ions and polar molecules cannot easily pass cross the hydrophobic core of the lipid, so they rely on the hydrophilic pores of channel proteins. Again, glucose can pass into the cell via carrier proteins because the larger polar molecules, glucose cross the membrane via facilitated diffusion, so they use carrier proteins. And then the oxygen statement passes freely through the membranes and it is soluble in lipids. So this is also correct because it lipid soluble, it diffuses quickly and freely directly through the phospholipid bilayer by simple diffusion method. Then the statement 4 is also correct. Glycoproteins act as antigens. Yes, this is also correct. On the extracellular surface, play a major role in cell-to-cell recognition and act as cells identify markers or antigen. So 4 statements are correct. So the answer is option A, because in answer A, all 4 statements are correct. Now moving towards the question number 20, and question number 20 is the diagram shows 2 cylinders of agar, and the agar cylinders are placed into solution of dye at the same time. The time taken for the dye to diffuse into the center of each cylinder is recorded. So this statement explains why the dye reaches the center of the one agar cylinder faster than the other agar cylinder. The dye reaches the center of X is the shorter time than the dye reaches the center of the Y because X has a larger surface area to volume ratio. The statement A is not correct because the reaches the center of Y faster, not X, because the surface area is larger of the Y, not A, because it is of small size. So statement A is not correct. Then the dye reaches the center of X in a shorter time than the dye reaches the center of Y because X has a larger, again, X has a larger surface area, not larger surface area. So again, this statement is not correct. Y has larger surface area. The dye reaches the center of Y in a shorter time than the dye reaches the center of X because the dye has a shorter distance to diffuse to the center. So the rules for process of diffusion are that the distance should be short. So this is the correct statement. Now statement D, dye reaches the center of Y in a shorter time than the dye reaches the center of X because Y has a smaller volume. Y does not have a smaller volume. The statement attributes the speed to smaller volume rather than the fundamental mechanism of a shorter diffusion pathway to core. So D is also not a correct option. Only C is there, which is the correct option. Now question number 21 is what are roles of adult stem cells in humans? Repair other cells by mitosis? No, they do not repair. Mitosis process of cell division that creates entirely new cells. It does not patch up or repair exactly existing damaged cells directly. Divide to replace cells and tissues? Yes, this is, it happens. This is the core function of adult stem cells. So they divide to produce new cells that differentiate and replace worn out or dead cells within a specific tissue, bone marrow, making new blood cells, skin stem cells, releasing epidermal cells. So in this way. Now the third statement, which is differentiate into new organisms, it does not happen. Adult stem cells are multipotent, meaning they only differentiate into limited range of cell types. Only a zygote has the potential to develop into an entire organism. So only statement 2 is correct, which is option D is correct. A, B, and C are not correct options. Now next question 22, which statements are correct for the mitotic cell cycle? DNA is replicated semi-conservatively during mitosis. DNA is replicated. This option or this statement is not correct. DNA is never changed from one. DNA is during mitosis. DNA replication occurs during the S phase of interphase. So which happens before mitosis begins. So this statement is not correct. DNA is never changed from one generation of cells to the next generation. DNA is never changed from this is also incorrect. Mitosis aims for genetic identity. The absolute statement never changes biologically false due to spontaneous mutations that can occur during DNA replication. So the mutations can change the DNA. Now, statement 3, the daughter cells at the end of a mitotic cell division have the potential to produce the same enzymes as the parent cells. Yes, it happens. This statement is correct. Mitosis preserves genetic identity. So, statement number 4, the same quantity of DNA is distributed to the nuclei of two new cells at the end of a mitotic cell division. The same quantity of DNA is distributed to the nuclei. Yes, this is also correct. During anaphase, sister chromatids are separated equally, ensuring both resulting daughter nuclei receive an exact equal amount of DNA. So, statement number 3 and 4 are correct. So, the D option is correct option. Now we reach at question number 23, and question number 23 is, you can see the photomicrograph shows a stage of the cell cycle. Which stage is shown in the photomicrograph? You can see this is anaphase. No, this is not anaphase, because in at anaphase the chromatids would be seen separating and moving apart into two distinct groups. So A is not the correct option. B is interphase. Again, interphase, they would not be, chromosomes would not be visible. So this is not the. Then metaphase. Metaphase, you can see they are arranged in the center. They are arranged in the center. So the metaphase is correct, because the photomicrograph classic squash preparation. So condensed distinct chromosomes are arranged in a circular alignment around the equator of the cell. So in this, it shows that this is the metaphase. Then telophase, again, they uncoiling into chromatin, they have reached to the opposite poles. So they get separated from each other. So in this way, they cannot be shown in this way. So C is the correct option over here. Question number 24 is there, and question number 24 is, the diagram represents a molecule of ATP. Molecule of ATP, J, K, and L. They are the components of ATP labeled J, K, and first of all, the J is, you can see the adenine base, adenosine triphosphate. So adenine base, so this is a purine. Nitrogenous base, which is characteristically made of a double ring structure. So this is purine. Double structure, because it has double structure. And then 6 carbon rings, you can see the 6, they are fused together. And K is sugar, which is have 5 carbon sugar. So this is pentose. And now these are the 3 phosphates. So in this way, the J is purine, K is pentose, and 6 is phosphate. So these D is the correct option over here. Question number 25, and question number 25 is which statements about complementary base pairing are correct? It occurs during translation. This is correct. Why it is correct? During translation, complementary base pairing occurs between the anticodon of transfer RNA and the codon of messenger RNA. So this statement is correct. Then purines and pyrimidines are the same size. No, pyrimidines and purines are of not the same size. Purines have double ring structure, whereas the pyrimidines have single ring structure. So therefore, they are not the same size. But the base pairs are of the equal length. This statement is correct because a complementary base always consists of one purine and one pyrimidine bonded together. So the total length of each base pair remains constant. So this maintains the uniform width of the nucleic acid structure. So this is the correct statement. Uracil forms 3 hydrogen bonds with adenine. No, this is incorrect because uracil pairs with adenine. So there are 2 hydrogen bonds between them. So only statement number 1 and 3 are correct, and it goes with the option B. Question number 26 is which statement is correct for protein synthesis? During translation, the strand of DNA used is the template strand. This is incorrect because template strand is used in the transcription, not in translation. So messenger RNA is formed when introns are removed from the primary transcript. In eukaryotic protein synthesis, the initial transcript contains both exon and introns. So post-transcriptional modification removes the non-coding introns and joins the exons together to form mature messenger RNA. So this statement is a correct statement. DNA primary transcript is modified by removing exons. So this is not correct because the primary RNA transcript is modified by removing introns, not the exons. Transfer RNA has codons which attach to anticodons on the messenger RNA. This is also transfer RNA molecules possesses anticodons which complementary base pair with the codons located on the messenger RNA strand. So this they have a wrong statement. So B is the correct option. So it was all about from question number 14 to 26. Please like, share, and subscribe my channel to watch more videos on these papers, or the third video of this paper will be published soon. Thanks for watching.